Welcome to our second podcast on bone. What we're going to deal with today is about how bone grows. We laid down the foundational components in our first podcast, the cells and the molecular components. Now we're going to put them together and see how the bone is able to grow during embryonic development, repair in both long bones and in our flat bones. So our objectives, we're basically going to compare and contrast the two types of osteogenesis or bone formation, intramembranous and endochondral. You're going to need to be able to identify the regions of the epiphyseal growth plate. These are going to be cartilaginous regions that we will distinguish histologically, and we'll look for each and every one of those. We're also going to need to list and identify the regions and compartments of long bones. We started touching on that a little bit before in the diaphysis and epiphyses. We will extend on that. And then lastly, we will describe the molecular processes of calcification. So during development, we basically have two types of bone forming. We have primary bone and secondary bone. Primary bone is going to essentially be calcified cartilage. That is going to take up, basically be a placeholder. And this is going to be where the bone is originally going to form. Osteoclasts will then come in, remove that primary bone, and lay down our adult secondary bone that we're going to maintain and modify throughout the rest of our adult life. So our two types of osteogenesis. Intramembranous, this is going to be de novo formation of bone from embryonic tissue. That means we're not going to go through any precursor cell types, any template types. We're going to start with mesodermal, mesenchymal cells, these pluripotential cells, and they are going to develop into osteoblasts and begin to lay down bone. Now to contrast that endochondral bone formation, we are going to first form a cartilaginous template of the bone. That cartilage template is going to calcify into primary bone. That's going to be removed by osteoclast. Osteoblasts are going to come in and lay down secondary bone, which is much more structurally tough for the remainder of our lives. Intramembranous is going to form our flat bones. Endochondral will be the type of formation we have for the long bones of our arms and legs. Now to start with intramembranous osteogenesis, it's fairly straightforward. We're going to first form a condensed core of mesenchymal cells somewhere during embryonic development. Those mesenchymal cells, as they condense, will differentiate into osteoblasts and begin to lay down both the organic and inorganic portions of the bone. At first, bone's going to be very, very small pieces, almost like little toothpicks, and those are called spicules. And as those spicules get larger and larger and as they coalesce into larger and larger masses, we then refer to those as trabeculae. That's the typical structure we see of spongy bone, even in our long bones. And that spongy bone, the actual bone itself, is considered to be trabeculae. Now, once again, these are all going to be derived from those mesenchymal cells, so we refer to those as osteoprogenitor cells, and they are going to continue to form more and more osteoblasts that when they become trapped inside the bone will become osteocytes. Here we can see we're just laying down bone layer after bone layer after bone layer. We've got our mesenchyme at the top going through these progressive stages, and here on the right you can see our different types of bone. Here on the right you can see our different types of bone. We have the outer table, we have the inner table, and then we have the spongy bone of our flat or intramembranous bones. Now, endochondral osteogenesis, this is going to be different. We have essentially two steps that we're going to go through. We're going to have a primary ossification center. Now, this is going to be that primary bone. We start out with hyaline cartilage. We're going to calcify that cartilage. Then we're going to remove it and place brand new secondary bone. So we'll have a secondary ossification center as well as we lay down that secondary bone. Now, our perichondrium around this cartilaginous template, that's where we find our blood vessels. We're also going to find blood vessels entering in through our periosteum. But this is where our chondrogenic cells are going to become osteoprogenitors. Remember, these are both mesodermal cells and these are pluripotential, so they can become either or. So chondrogenic cells become osteoprogenitor cells. Epichondrial cells become periosteal cells. And so we're just transitioning that cartilage into bone. This gives you an idea of how this takes place. We have our cartilaginous template that's hyaline cartilage, and the blood vessels in the center of the diaphysis of this template. These blood vessels are going to come in. We're going to begin to break down that cartilage. You can see what's happening here. And the yellow that you see, we are transitioning that into first primary and then secondary bone. And we continue to extend this. This is our primary ossification center. In many instances, the epiphyses of our long bones are considered secondary ossification centers, and they're going to go through that same primary then secondary bone. And we're slowly forming the marrow cavity. We're slowly forming our compact and then spongy bone until finally we get to a state where the only cartilage we have is going to be at our epiphyseal growth plates on each end, and we still have the articular hyaline cartilage on our articular surfaces of these bones. Here you can see that cartilage here in blue on the very tip, and we can see the growth plate cartilage existing in our epiphyseal plates. Now, the way that that hollow cavity begins to form first, we have a fairly dense matrix of cartilage to begin with. Well, the osteoprogenitor cells that are present at that central part of the diaphyses, they're going to start forming that compact bone first. And it is going to shut down the diffusion of materials, because remember, that's how chondrocytes receive their nutrients. And as you shut down the diffusion, because you're creating a sub-periosteal bone collar, you're essentially choking off the chondrocytes, and the chondrocytes die in the core. And then the vascular buds that are coming in from that periosteal layer, invading into the space, they're bringing in the osteoprogenitor cells that are going to be laying down the secondary bone. They're bringing in also stem cells for hematopoietic tissue, the red cells and the white cells, and also blood vessels entering into that area. So once we bring in the osteoprogenitor cells into these cavities, then we're going to have the osteoprogenitor cells laying down that calcification layer. So it's going to be calcified cartilage, and it's going to appear that dark purple. We saw that in a slide when we looked at the other bone lecture. Well, then from that calcified cartilage, osteoclasts come in. They remove that primary bone, that calcified cartilage, leaving a space that then osteoblasts can come in and begin to lay down that true, pure bone matrix of secondary bone. So here we see our cartilaginous template, the perichondrium here. Now we can begin to see that first compact bone coming in, and as we do so, we're choking off the chondrocytes in the center, creating this hollow space. Blood vessels come in, bringing our osteoprogenitor cells, and we're converting this into first primary bone, which is that calcified cartilage. That's removed by osteoclast. Osteoblasts then come in, again along with the blood vessels, to lay down the secondary bone matrix. Now, the secondary ossification center, if we could go back just quickly, you can see in the epiphyses, blood vessels are also growing into the epiphyses. We're doing very similar things in the epiphyses, so that really we're going to have bone on bone, the center's bone, with two layers of these hyaline cartilage in our growth plates. Now, as we lengthen our long bones, this is going to happen in a couple of different ways. One way is we can have interstitial growth, and this is growth in the epiphyseal growth plates, and we're going to look at various regions of how we do that. But we're also going to have appositional growth that's coming in from the area of the diaphysis as we thicken the bone in that area. But this interstitial growth at the epiphyseal plates, we're going to have proliferation and mitotic activity in the hyaline cartilage in those epiphyseal plates. That's going to lengthen the bone, and then we will gradually replace that cartilage. We'll calcify it. Osteoclasts will come in and remove it. Then we'll lay down osteoblast and secondary bone, and that's how we will lengthen the bone. So in our illustration here, we can see our epiphyseal growth plate, and we can see how this color along the middle is much more indicative of what we see with cartilage than with this more eosinophilic look that we have to our bone matrix. And so we're going to identify different regions within this epiphyseal growth plate. So first, we're going to look for different zones. We have about 5 zones that we're going to identify here, and we're going to start with the zone of reserve cartilage. This is where we're going to basically have what looks like hyaline cartilage. We'll have our isogenous groups. It's not basically going to look any different than we would see with our hyaline cartilage. But then all of a sudden, we are going to see the zone of rapid proliferation where isogenous groups, the chondrocytes, are going to get so crammed together that they're going to look like stacks of coins stacked up against each other and lined up. That's going to indicate we are in the zone of proliferation, very rapid growth that we see there. Then our lacuna are going to absolutely enlarge. The cells are going to enlarge, the lacuna are going to enlarge. There's just a lot of white space that we begin to see. This is going to be the zone of maturation and hypertrophy, this tremendous growth of the lacunar spaces of our cartilage. So here in our illustration, we can see bone, this calcified cartilage we see here in the purple. Then our bone is going to be this reddish color. We see our osteoprogenitor cells here on the far right. As we move back and look in our epiphyseal plate, we can see hyaline cartilage very clearly in this region. This is our zone of reserve cartilage of our epiphyseal plate. Then we get into this zone where we begin to see these stacks of coins. This is going to be our zone of proliferation. You can see these stacks real nicely. We have another image coming up where you can see those. And then that transitions into this area where we see this tremendous amount of white space, maturation and hypertrophy. So this is the hypertrophic zone, this is the proliferative zone, and this is going to be the reserve zone. Now looking a little bit more closely, here you can see just the stack upon stack upon stack of our chondrocytes. This is the proliferative zone in our epiphyseal plate, and all of the white spaces that we see, this is going to be the zone of hypertrophy and maturation zone. We're making a lot of space in these lacuna as we lengthen these bones, and now we're going to begin to calcify that in our next region that we see at the bottom of this particular image. So adjacent to the zone of hypertrophy, we have the zone of calcification. This is that calcified cartilage. As the chondrocytes are dying, they're leaving behind that matrix. Cells are coming in and laying down that cartilaginous component. Then in the last final zone, this is going to be continuous with our marrow cavity. This is going to be the zone of ossification, and that's where we get our secondary bone laid down. Osteoprogenitor cells are coming in and building that truly lasting bone matrix. So once again, a nice diagram showing the different zones that we have within our epiphyseal plate. Down at the bottom, we have our, here we go. We've got our cartilage here at the top. They call it the resting zone. That is going to be our reserve cartilage. Stacks of coins, that's our proliferative zone. Hypertrophic zone. Then we get our zone of calcification. Calcification then is going to lead into where we see forming trabeculae. That's going to be our secondary bone, continuous with our marrow cavity. And we'll see more of those images when we get into our laboratory discussion. Now, when we look at the activity of our epiphyseal plate, you have to understand that the lengthening of the bone is going to be the net result of bone formation and bone resorption, osteoclastic versus osteoblastic and osteocytic activity. So as long as the resorption equals proliferation, we're good. However, once we get into the early 20s, that's when proliferation stops, but resorption continues to progress. And so as we do that past about 20, 25, we lose that cartilage component of our epiphyseal plates, and our bones will be unable to continue to lengthen anymore. We will have reached our full length and size of our long bones in our body. So we talked about interstitial growth. Now with appositional growth, instead of lengthening the bone, we're widening the bone. And with appositional growth, we're going to be looking at osteoprogenitor cells and osteoblasts in the periosteum rather than that in the epiphyseal plate. This is going to be used to thicken the bone, especially that compact bone, because when you think about it, we're hollowing out the marrow cavity, so structurally it's not going to resist those compressional forces and those vectoral forces trying to make the bone go laterally. think about it, as your body pushes down from gravity on your legs, they're wanting to expand laterally. So we need that extra structural support so we can expand the marrow cavity where we're going to have hematopoietic tissue and fat storage in those areas. So we've got to have this appositional growth mediated and facilitated in the periosteum. Now, whether we're talking about appositional or interstitial growth, anywhere we're calcifying cartilage or laying down that proper secondary bone calcified matrix, this is going to be initiated through essentially what is referred to as a heterogeneous nucleation event. We're basically going to form these little crystals, and they're going to be niddies of crystallization. I think that's on the next slide. It's going to be where we begin the process of forming these crystals. And basically, as we produce these materials inside the vesicles of our osteoblast, they're going to be released into the microenvironment surrounding the cell, because we can't have these crystals forming inside the cell. That would kill the cells. So these vesicles that are going to release this material into this immediate extracellular microenvironment, we got calcium phosphate, cyclic AMP, ATP, because we do have ATPase activity in many of these enzymes. We've got alkaline phosphatase, pyrophosphatase, calcium binding proteins. All of these materials are going to be used to begin to form those hydroxyapatite crystals. Now, in addition to all of those materials, we're going to have calcium pumps. They're going to pump more and more calcium into these vesicles, and these vesicles are going to float out into this microenvironment. Remember, we're not doing this inside the cell. So as we enrich the calcium with all of the materials present needed to form these crystals, we are going to create the crystals. They're going to begin to form, and they're going to rupture through the membrane, and that's going to be the start of this hydroxyapatite crystal out in the matrix. And again, it's going to start as a seed. You're going to seed with these little crystals called niddies of crystallization. They're going to expand, 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 and they're going to coalesce together into what we know as our calcified bone matrix.